Okay, hopefully everybody can hear me okay. This is kind of a new format, but it seems to be going well so far. First off, it's an honor to be presenting at this year's Earl Davie Symposium, and I really regret that I cannot be there in person today to share all these fun presentations with you. So before I begin, I'd also like to preface that with respect to today's presentation, I will not be elaborating on an abundance of data, just because we are in the process of filing some IP and there's an ongoing disclosure. So with that, I'd like to begin by introducing myself. I'm Colton Strong. I'm currently a second-year master's candidate, currently co-supervised by Dr. Christian Kastrup and Dr. Dana Devine. And this is my project on enhancing transfusable platelets using mRNA therapy with the goal of producing exogenous proteins. And to begin, I'm sure you all are relatively familiar with severe hemorrhage and major trauma, which is a major cause of death for both civilians and for soldiers and military personnel on the battlefield. Bleeding and associated morbidities is associated with approximately 40% of surgical deaths, and uncontrolled bleeding is the leading cause of potentially preventable deaths in young people worldwide. And in the military theater, not surprising, bleeding, unfortunately, is the #1 cause of preventable military deaths. And so despite, although there's been dramatic improvements into platelet storage handling practices, blood product inventory management, there remains to be immediate innovation on the transfusables themselves. And this really highlights that new transfusion technology and innovative approaches are really needed to better control bleeding in the future. And so to position my project a little bit more adequately, I'd like to go over some of the current hemorrhage management practices. And this typically begins when a patient or a military personnel enters the trauma care center, and there's the identification of massive hemorrhage. This is usually characterized by 50% blood loss within 3 hours and dramatic bleeding. And so once there's been the identification of massive hemorrhage, there's the activation of what's called a massive transfusion protocol. And this generally consists of the administration of transfusables, including plasma, packed red blood cells, and platelets at a 1:1:1 ratio. And hopefully that is enough to control bleeding. So focusing more on the platelets, their role during transfusion and in general circulation is they detect and adhere to sites of vascular damage. And this is where they enable the localization of the coagulation response and the coagulation cascade, in addition to interacting with neighboring platelets to generate that initial platelet plug, and really are responsible for releasing key mediators during activation that can not only drive coagulation in the formation of the clot, but also can modulate tissue remodeling and a return to homeostasis. So with hemostasis, my mistake. So with that, during severe hemorrhage, however, there can actually be some complications that can arise if there's been too much blood lost, so hypovolemia. And so this can result in what's considered the triad of death, a very scary nomenclature, which consists of acidosis, hypothermia, and rendering the coagulation cascade and all of the associated proteins less functional and resulting in impaired ability to generate a clot. And one of the negative effects of this is that this triad of death and acidosis can really impair platelet function and activation. And so what happens is during a trauma setting, transfusion setting, the transfused platelets, for example, may have impaired activation and no longer be able to control bleeding. And this can lead to patient exsanguination or death by blood loss. And so one of the core questions of this project and actually of our lab in general is, can donor platelet function actually be enhanced? And so the answer to that question is yes, indirectly at this point. And so one of the interesting studies that came out of the BRI, the Medical College of Wisconsin, was in a study where they looked at genetically engineering megakaryocytes, which are the platelet precursors, using a viral transfection protocol with the goal of engineering megakaryocytes to produce factor VIII. And then they took these modified megakaryocytes and transplanted them into a canine model of hemophilia A, which is factor VIII deficiency. And what they're able to show is not only is factor VIII produced in these modified megakaryocytes in the bone marrow, the factor VIII is also localized to platelets, and they are able to show that this intervention actually can ameliorate blood loss and can actually restore clotting abilities. So this highlights that platelets would serve as a logical target for any sort of gene therapy. But the next question is really are anucleate platelets, so platelets that lack a nucleus, are they able to be amenable to RNA therapy? And so in an older study, we have been able to see that platelets after storage do express and produce new proteins. So in this study, they are looking at GP3A, and they show that there is de novo synthesis of these proteins during storage. And so this really highlights the core hypothesis of our project, which is that we can leverage lipid nanoparticle technology to load hemostatic or therapeutic mRNA into these lipid nanoparticles and then modify donor platelets with the intention of expressing these therapeutics or hemostatic targets and endow these new modified platelets an enhanced ability to stop bleeding. And so just to go over a little bit about lipid nanoparticles, I'm sure you're all very aware about this technology now, especially after our the exciting therapeutic success of Pfizer and Moderna COVID vaccines. But that's not to forget the first siRNA LNP that was approved on Patro. And so using a similar formulation, and this is where I can't go into too much detail, but we're using this technology to deliver mRNA into our platelets. And so the three objectives of my project really are to, one, achieve the expression of therapeutic protein targets in platelets. Number two is to determine the in vitro coagulability of LNP modified platelets. And number three is to assess the in vivo coagulability of these LNP modified platelets in rat model of polytrauma. And so the data that I can show today is pretty exciting. In one study done by Jerry Lung, a current graduate student, he was able to show that using donor platelets, you could take LNPs and take the washed platelets and treat them with a dye labeled LNP, so the actual fluorophore within the LNP. And then after 24 hours of treatment, assess GFP expression, look at LNP uptake and internalization, and look at a platelet marker all on flow cytometry. And the data that is shown on the right indicates that as you increase the dosage that you treat these platelets at, so increase the amount of LNPs per platelet, you do see that an increase in the dye fluorescence intensity, which is indicative that these platelets are uptaking these LNPs and internalizing them. But the big question is, so you get the mRNA, and now what? Additional data from this study has indicated that we do see expression of GFP, for example. So when the dye labeled LNPs contain mRNA encoding GFP, after platelet treatment for 24 hours, you do see an increase in the number of platelets that are considered GFP percent positive. And so this is a pretty exciting data that's really been the foundation of my project and other ongoing projects in our lab. And so just to kind of summarize, so currently we're really optimizing this platform to maximize the expression of exogenous proteins and also develop systems in which we can minimize the effects of lipid nanoparticles on platelets to really try and deliver exogenous mRNA with the least amount of perturbation to any sort of platelet physiology. So once we kind of finish optimizing this platform, we want to identify the consequence of LNP treatment on platelet function using a number of standard characterization assays, including rotational thromboelastometry, and then also light transmission aggregometry, which will give us an indication of the effects of these LNPs on platelet to platelet interactions, and with ROTEM giving us an indication of how modified platelets might be changing clot formation parameters and dynamics. And so with that, I'd like to thank everybody in the Castrop and Divine labs, especially Jerry Lung, Dr. Katie Badior, and Madeline Robertson, and then also everybody in the Divine lab who has been incredibly welcoming, and all our collaborators as well, and additionally all of our funding sources. So with that, I'll take any questions, and thank you guys for listening.
Thanks very much, Colton. Without giving away any trade secrets, what sort of proteins would you be focused on getting platelets to express so that you could enhance the coagulation system?
Yeah, that's a very good question, and I'll be a little bit vague, but our current target, we're really focusing on two kind of classes, actually three classes of targets. One would be small peptides that we could easily express in platelets, and then others being larger, maybe coagulation factors or therapeutic proteins that we could then express in our platelets, and then hopefully achieve localized delivery of these coagulation factors, of these therapeutic proteins, directly to the site of injury. But we're also looking at a bigger picture. We're hoping this can be a platform technology. And one of the most exciting things personally is that if we are able to demonstrate reliable expression of any target protein, this would enable platelets as a cell therapy in a variety of clinical indications. And really establish a plug and play system where you can express your protein and deliver it in cancer or in inflammatory diseases, stuff like that, where platelets have a more and more are becoming there's a role of these platelets in all of these different diseases. So we're pretty excited about that.
Thank you. Looks like we've got a couple questions in the back.
Okay, that's fascinating. So you're going to try and sort of ramp up a variety of platelet contents with a certain amount of energy available in the platelets and a certain number of amino acids. Are you going to get into a competitive situation as you try and put in multiple stimulus?
Yeah, I think you bring up a really good consideration that we have, and that is we're really trying to leverage platelet machinery to produce exogenous protein. However, there is a competitive nature to it where if there are proteins that are being expressed, then we might not necessarily be able to generate enough protein or significant levels of protein. So that's actually a really active question that we're trying to investigate is how do we design our mRNAs? How do we design our LNP formulations where we can promote the expression of our delivered mRNA over any sort of competing mRNA sequences present? And so that is going to be a challenge that we're going to have to overcome is expressing physiologically relevant levels of protein.
Can you hear me? Okay. Nice talk. You might not be able to answer my question in too much detail, but can you maybe give the broad picture of how you're targeting these lipid nanoparticles to platelets as opposed to other tissues like the liver, which is really good at taking up lipid-based particles?
Yeah, I'd be happy to comment on that. And I apologize if that wasn't clear in my presentation. Our current source of platelets and the current application of this technology is in an ex vivo system. And so we're working with platelets that have been retrieved from donors. And so these platelet units contain the platelets of 4 donors that have been pooled into a unit that would be then transfused into a bleeding patient. So we're not actively trying to deliver our LNPs to platelets in vivo yet, but right now we're working on delivering them outside of the body. So that's why we are able to target them and target platelets without seeing any sort of off-target localization to the liver.
I have a question. Sorry. You mentioned some potential consequences of LNPs on platelets, and I was wondering if you could maybe just expand a bit more on what you would expect as consequences, because you mentioned that you were going to be investigating them, but what are just some possible things that you were thinking about looking at?
Yeah, this is a really new space, and so it's really an unknown about what the effects are of lipid nanoparticles on platelets. We do know that platelets are highly sensitive to environmental perturbations. So any sort of shaking or contact activation, these platelets can undergo changes in morphology. So we're certain that the LNPs are having some degree of an effect on the platelets. The question is that render them non-functional, or are we able to? And the goal of the project is to deliver this these mRNAs without actually dramatically changing any sort of platelet physiology. So we're going to be looking at a number of indications of platelet activation, changes in morphology, changes in ability to initiate and generate a clot, and that'd be through rotational thromboelastometry, and then also ability of these platelets to aggregate and to bind with one another through that fibrinogen bridge to form that platelet plug. So very good question, though.
Hi. Okay, good. Last question before we break. I kind of wondering, great talk, by the way, really fascinating. How is the practicability of getting the LNPs into platelets? So in this case, if you have massive transfusion, how long is it going to take? And then to transfuse into the patient, because it's probably, you need less than an hour to do all of that.
Yeah, also a great question. It's something that the team and I are always asking ourselves is what is the clinical feasibility of taking platelets, modifying them, and then transfusing them into a patient. And so when without giving away too much, we are getting some really exciting results about how we can make this a clinically relevant practice and how we can deliver mRNA into these platelets in a clinically relevant time scale. So, and then take advantage of some of the innate practices that are involved when it comes to taking a platelet unit from storage and then transfusing into a patient. There's a number of steps that we can integrate our technology into, I believe, pretty robustly. And so but it is something that we have to keep at, making sure that we keep clinical relevance in mind as we do this.
Much, Colton. We'll let you go back to whatever you're doing in Wisconsin.